So we have already started with the amino acid chapter, proteins and amino acids. We'll see the remaining points. This chapter is very lengthy chapter. Proteins and amino acids simple chapter nahi hai, खूप lengthy chapter hai. It is very lengthy chapter, and it will take a lot of times. If you want to explain each and every single thing properly, it will take around 8 to 10 days, because proteins and amino acids not an easy thing. Okay, but I will explain you whatever I can, whatever is important for your GPAT and other exams. Okay, so we have already seen this part. What do you mean by proteins and what are the basis of protein? So remember this point. Kage-Dahl method is used for a measurement or estimation of nitrogen. Kage-Dahl method. So there was a question on this Kage-Dahl's method. It is a method used to determine the nitrogen content in the organic compounds. So proteins are the polymer of L-alpha amino acid. You have to remember 20 standard proteins are there. 21 is a cysteine and pyrolysin is 22nd. We have already seen. We have already seen the function of protein, static function, dynamic function. So we have seen the composition of protein also, as far as the elemental composition of proteins are concerned. We have seen the basic structure of amino acids. We have seen this part. So classification of amino acids based upon the nutritional requirement, classification based upon the polarity, classification based upon the source. So whether the whatever skeleton is remained after metabolism, that skeleton is used as a precursor for production of sugar, then it is a glycogenic, and if it is used for the production of keto acid, then it is a ketogenic. We have seen the structural classification of amino acid also. So first class was amino acid with the aliphatic side chain. So in that you have Gauri, glycine, alanine, valine, leucine, isoleucine. Second class was a second class was the amino acid containing OH group, that is hydroxyl group. So short take is nothing but the ST. So S for serine and T for threonine. The third class is nothing but the sulfur containing amino acid. So in that you have C-squarium, cysteine and cysteine. Cysteine and cysteine. Cysteine is having sulfhydryl group, cysteine is having disulfide bond, and methionine thiourether. So after that, acidic amino acid and their amides, aspartic acid, glutamic acid, and their respective amines. So aspartic acid, glutamic acid, aspartamine, asparagine, and glutamine. Then then we have seen the basic amino acid lysine, arginine, and histidine, LAH. Then aromatic amino acids we have seen phenylalanine, tyrosine, and tryptophan, all the aromatic amino acids. So the ring percentage is nothing but the benzene or phenol and indole respectively. Then last amino acid is known as a amino acid. Last amino acid is known as a amino acid. It is not a proper amino acid. It is known as an amino acid because there is an imine group. That's why proline is an amino acid. So here pyrrolidine ring is there. Remember, we have seen the structure, general structure, the isomerism in the amino acids. We have seen the amino acids. They are having two groups. Okay, ammonia, there is amine group and carboxylic group. So most of the amino acids they exist in a body in the form of ions. Okay, so this NH3 plus is responsible for the basic nature of your amino acid, and COO minus is responsible for the acidic nature of your amino acid. So it acts as a buffer, but there is one pH, one specific pH at which amino acid remains as a bipolar or dipolar or hybrid ion. It is known as a deuterion. Okay, deuterion. The pH at which amino acid exists as a bipolar ion or dipolar ion or deuterion, known as isoelectric pH. So every individual amino acid has its own isoelectric pH, and there is a list given in the book. If you want to see, you can see. Otherwise, I will also copy that list from Satyanarayana and other books, and I will teach you. So as that of the amino acid also has an isoelectric pH. तो वैसे जसं अमाइन ऍसिडचं असतं तसंच आपल्या प्रोटीनचं पण असतं. सो प्रोटीन ऑल्सो हैज अ आइसोलेटेड पीएच. ओके, सो वेरी सिंपल एग्जांपल, आप मैं एक कोट करता हूं आपको. सो ऑनगोइंग है तो you might have seen the milk. So milk is nothing but the containing protein. So in that milk you have casein. Okay, and now what happens, अगर मिल्क दूध जो होता है, दूध आहे तो दूध काय होतं, फाटतं. कधी फाटतं? when there is a microbial growth, if there is a contamination in the milk. त्याच्यामुळे so if there is a contamination, that means so microbial, what they do, they decreases the pH of milk. Okay, so when pH will come around 4.6, 4.5, 4.6. That is the isolytic pH of your casein. Casein is nothing but the milk protein. So at that isolytic pH, so proteins, they get precipitated, they get coagulated. That's why milk will coagulate. That's why the part of okay. So that's why acidic pH. Okay. So we have seen the taste, optical rotation, amino acids as useful as a drug. We have seen this part. We have also seen the reactions of amino acid due to reaction due to carboxylic group, reaction due to amino group. Okay. Identification of amino acid is done by these reagents. Sagnus reagent is nothing but the one fluorophore and nitrobenzene. You have to remember this reagent question. Reactions with the densyl chloride and Edman reagent is phenyl isothiocyanate. So these reagents, they are used to determine the amino acid sequence in the protein also. We have seen these reactions. After that, we have seen the color reaction. You have to remember biuret reaction is given by a protein containing at least two peptide linkage. जिसमें दो पेप्टाइड ग्रुप होगा, वही क्या देगा, बायोरेट रिएक्शन देगा. बायोरेट मतलब बाय मतलब टू, यूरेट मतलब क्या, यूरिया. When two urea molecule will combine, will condense at a 180 degrees, so there is a formation of biuret. Now this biuret react with the copper sulfate to form a biuret copper complex which has a particular color. Now ये biuret बनता कैसे, when two proteins, when protein containing two peptide bond linkage, so when it is treated with the sulfur, it is sulfur, cobalt sulfate, sorry, copper sulfate, and then there is a formation of biuret due to two urea group of protein, and that's why for a biuret reaction two peptide linkage is important. Question इसके ऊपर आ चुका है. In nitrogen reaction is given positive by alpha amino acid, xanthopatic for aromatic amino acids, mylose for phenolic group of tyrosine. Hopkins Cole reaction is given positive by indole ring present in the tryptophan. Sakaguchi reaction is specific for gonadino group of arginine. Nitroprusside reaction is for sulfur group of cysteine. Again, sulfur test also for same thing. Pauli's test will be given positive by imidazole ring containing amino acid, that is H3. इसके ऊपर भी क्वेश्चन आ चुका है. Follin कोई कैल्टोस टेस्ट given positive by phenolic group of tyrosine. Okay, so we have seen the decarboxylation of amino acid result in the respective amino, and transamination means transfer of amino group. Okay, transfer of amino group from one amino acid to the keto acid. Okay, so this one is known as a glutamate, and this one is known as a pyruvate. So pyruvate is nothing but the keto acid, and this glutamate is nothing but the amino acid. Now what happens? So this amino group is transferred to a pyruvate, and which will produce you alanine. Now this glutamate will be converted into another keto acid known as alpha ketoglutarate. Now this alpha ketoglutarate, you know that it is intermediate in the Krebs cycle. So to produce the respective new amino acids, so transamination reaction is very important. It has a very important role in the protein metabolism, transamination and deamination. Protein metabolism में वही दो reaction होता है, transamination and deamination. Now these two reactions are very important. After that, we have seen the how there is a formation of disulfide bond, how there is a formation of peptide bond. Okay? So when amino group of one amino acid, react with the carboxylic group of another amino acid, so there is a formation of CONH bond, which is known as a peptide bond. Okay, peptide bond formation, right? Now this is known as a monopeptide. A monopeptide will have two amino acid and one peptide bond. A dipeptide will have three amino acid and two peptide bond. A tripeptide will have four amino acid and three peptide bond. Likewise, a decapeptide will have 10 peptide bond and 11 amino acid, like that. Okay, so you have to be clear about that. I hope I am audible to you clearly. Yes or no? Now after that we have seen amino acids and their neurotransmitter respective derivative. Glycine neurotransmitter होता है, glutamate भी होता है, ये अपने already देखा है, so we will not take this. So few biogenic amines and their function we have already seen. So serine after decarboxylation you will get ethanolamine. Ethanolamine is a storage form of choline and also required for the phospholipids. Then glutamate. So after the decarboxylation of glutamate, so you are going to get glutamic acid. So there is a gamma amino butyric acid. Okay, from the glutamic acid only GABA is synthesized, which is inhibited neurotransmitter in the brain. From histidine, decarboxylation gives histamine, which has a physiological role. From phenylalanine, phenylalanine से बनता है डोपामाइन, सॉरी phenylalanine से बनता है टायरोसिन, टायरोसिन से बनता है डोपा, डोपामाइन से डाइहाइड्रोक्सीपhenylalanine, डोपा से बनता है डोपामाइन, डोपामाइन से फिर बनता है आपका नॉरएड्रेनालिन एंड लास्टली एड्रेनालिन. ठीक है. So again it is a neurotransmitter for sympathetic nervous system and other. टायरोसिन से बनेगा टायरामाइन, ट्रिप्टोफैन ट्रिप्टोफैन से बनेगा ट्रिप्टामाइन, सेरोटोनिन एंड मेलाटोनिन. Melatonin and melanin, there are two names, melatonin and melanin. Melanin जो होता है that is a pigment present in the skin responsible for the color of skin. Melanin is a different. Melatonin is a hormone. Melatonin is a hormone responsible for the circadian rhythm. Then cysteine, after decarboxylation you are going to get taurine, which is a content of bile. Now we already seen this non-protein amino acids, so I will not repeat it again. Okay, non-protein amino acids we have already seen. Now structure of protein. So what is protein? Protein are nothing but the polymer of L amino acid. So proteins are formed by amino acid. When amino acid will form a peptide bond and this repeating unit of amino acid in a peptide. Okay, what is peptide? Peptide normally, peptide is the term which is denoted for a sequence of around 10 to 20 amino acids. अगर 10 amino acid का sequence, 20 amino acid का sequence, if there are peptide bonds, so it is known as a peptide. Okay. Now polypeptide, more than 50 amino acids, right? Now protein is there when the amino acid sequence is more than 200 amino acid sequence is there, then it is known as a protein. Okay. The normally term protein is applied. Nowadays it is applied for a simple peptide also, but normally protein is the term when there is involvement of two or more polypeptide. Okay. Protein are formed by amino acids by peptide bond. Amino acids will form a, okay, two amino acids will form a one peptide bond. When two amino acids they will react with each other, the amino group of one amino acid react with the carboxylic group of another amino acid. Then you are going to get a peptide bond. Now a peptide bond is formed between two amino acids. Remember, two amino acids will form a one peptide bond. 3 amino acids will form a 2 peptide bond, and likewise more than 50 amino acids will form a single peptide. More than 50 amino acids will form a single peptide. Amino acid sequence can be, amino acid sequence in the protein can be analyzed, can be determined, can be sequenced by reagent. So first reagent कौन सा है? Sanger reagent. Sanger reagent known as a 1-fluoro-2,4-dinitrobenzene. Sanger reagent के ऊपर क्वेश्चन आ चुका है, 2 बार हो चुका है ये रिपीट. Sanger's reagent, so 1-fluoro-2,4-dinitrobenzene, dinitrobenzene, है ना, fluoro, okay, fluoronitrobenzene, DNB, FDNB short. Then Edmonds reagent is there, known as a phenyl isothiocyanate, and then densyl chloride is also there. So there are different agent which will bind to a particular terminus. They will, both Sager's reagent and this Edmonds reagent, they will bind with the amino terminus. जो भी अमीनो एसिड का अमीनो ग्रुप होता है वहां पे जाके ये रिएजेंट बाइंड करते हैं, है ना, और वो अनदर कंपाउंड तैयार करते हैं. उसके बेस पे यू कैन एनालाइज कि सीक्वेंस क्या है अमीनो एसिड. मतलब अगर कोई पेप्टाइड है 50 अमीनो एसिड का, तो पहला अमीनो एसिड कौन सा है, दूसरा कौन सा है, तीसरा कौन सा है, ऐसा यू कैन एनालाइज. दैट इज sequenter. Okay? You can analyze the sequence of protein by 2-3 reagents. Sager's reagent, then Edmonds reagent, then densyl chloride is also there. I hope I am audible to you. Am I audible? Am I audible to you? Okay. Now structure of protein. Protein, you know that protein is nothing but the polymer of L-alpha amino acid. Repeating monomer amino acid. Now structural hierarchy of protein. Now structure of protein may be divided into different types. Okay. A primary structure of protein, secondary structure of protein, tertiary and quaternary structure of protein. Primary structure of protein means what? तो primary structure होता है वो क्या होता है? Primary structure means simple peptide linear chain. Simple linear peptide chain. Now what happens in that? So when two amino acids will come together to form a bond, that is a CONH bond known as a peptide, monopeptide. 3 amino acid will convert, will 3 amino acid will in a dipeptide, like that. So CONH bond, that is a peptide bond, a linear. The primary structure of protein is a linear in the nature. Primary structure of protein, it start from N terminal to the C terminal. Okay. Now measurement of protein structure or reading of peptide is done from N terminus to the C terminus. मतलब N terminus मतलब क्या, एक अमीनो एसिड का अमीनो टर्मिनस, NH2 जो होता है उसको अमीनो टर्मिनस बोलते हैं, NH3. So you have to read from that amino terminus to the carboxy terminus of last amino acid, because amino acid okay, protein synthesis also takes place from N terminus to the C terminus. Okay. Now protein synthesis is controlled by your genetic material, DNA. So whatever nucleotides present on the DNA, so that sequence will be read by mRNA, and according to that, okay, according to the code, according to the genetic code, the protein will be decided. According to the genetic code, the amino acid will be added, because you have different genetic codes for 20 amino acid. 20 amino acid के लिए different genetic code होता है. Okay, 2 amino acid will form a 1 peptide bond, 3 amino acid will form a 2 peptide bonds. This one. Now this is known as a primary structure. The primary structure is nothing but the having this bond. Primary structure में कौन सा bond होता है? CONH. So in the primary structure, peptide bond is there. You have to remember. Question आता है इसके ऊपर. Primary structure में कौन सा bond है? Secondary में कौन से bond है? Tertiary में कौन से bond है? इसके ऊपर question आता है. So in the primary structure, which is linear, just there is a peptide bond, CONH bond, which is a linear structure. Okay, right? Now secondary structure of protein. Secondary structure of protein. Secondary structure of secondary structure of protein. So in the secondary structure, why secondary structure is there? So now in this primary there is a single peptide. Now in the secondary structure, this peptide may turn, or there is an interaction between hydrogen bonds. Interaction between hydrogen bond, or two peptides, if they comes together, there is an interaction between hydrogen bond. Okay, so hydrogen bond of amino and COH of carboxy. So if there is interaction between two peptide chain or a simple peptide, folded peptide, you know, तो उसमें जब interaction होता है, when there is interaction hydrogen bond, when there is a hydrogen bonding between two peptides, or is hydrogen bonding between the same peptide which is folded back. Okay, so this is nothing but the twisting. So secondary structure of protein is because of the twisting of your primary structure. The primary structure linear structure होता है, उसका twist अगर करेंगे तो secondary structure it will give rise to secondary structure. So in the secondary structure you have hydrogen bonds. You have to remember, in the secondary structure you have hydrogen bonds, right? Secondary structure of protein, that is hydrogen bonds of different amino acid. A hydrogen bond between amino group and this carboxyl group. Okay, when a protein will twist. Okay, now in the secondary structure you have, it is largely determined. It is there are two models for this. Okay, first one is known as alpha helical structure. Alpha helical structure. Okay, Pauling. There was one scientist known as Pauling. He depicted this alpha helical structure. Okay, so alpha helical structure and beta pleated sheet. So there are two structures in the secondary structure of protein. Alpha helical structure. First one is alpha helical structure, and second one is nothing but the beta pleated sheet. So what are the structural feature of alpha helical? तो alpha helix में क्या रहेगा? Secondary structure of protein, ठीक है? It is made by twisting and turning of your simple peptide. There is an involvement of hydrogen bond. Involvement of hydrogen bond. आप बोलेंगे सर यहाँ पे सिर्फ हाइड्रोजन बॉन्ड रहेगा, नहीं, जो पहला बॉन्ड है CONH वो तो रहेगा ना, इसमें ऑलरेडी वो है. In addition to that, it has a hydrogen bonding. Now there are two simple structure in the secondary structure of protein. So alpha helical structure. Okay, so it is made by, as the name indicates, helical. Okay, so what is helix? So COH bond will be the backbone of that helix, and amino acid will be in the surrounding. That's why it will look like a helix. That's why it is known as alpha helical structure. Now in the each turn of alpha helical structure, you have a 3.6 amino acids. इसमें एक जो टर्न होता है, I will show you the diagram also. So you have 3.6 amino acid. Now distance है ना covered is a 0.4 nanometer. Okay. Now okay. So in each turns of each is in loop of each in a one loop of alpha helix you have 3.6 amino acid, and two loop between distance between two loops, that is a 0.4 nanometer. Now there are two types of alpha helix, right handed alpha helix and left handed alpha helix. दो alpha helix होता है right handed alpha helix, okay, and left handed alpha helix. So right handed alpha helix is a more stable and it is a more common the protein. तो right handed alpha helix more common होता है और it is a stable होता है. So right handed alpha helix is more stable and common in the protein. It is more stable than left handed helix. The amino acid proline disrupt the helical structure. तो जो secondary structure होता है that is disturbed by amino acid proline because it is an imino group. It is having imino group. So proline will disturb the proline will disturb the alpha helical structure of protein. ठीक है? Not only proline but some other amino acids like for example acidic amino acids, then basic amino acids. So acidic amino acids and basic amino acid proline they will disturb the structure of your alpha helix. जो alpha helix का structure होता है उसको disturb कर दे ये proline and acidic amino acid and basic amino acid. दूसरा एक secondary structure होता है उसमें beta plated sheet. So it was again proposed by Pauling and Corey. So these are the two scientists who demonstrated the primary secondary structure of protein, Pauling and Corey. So they only mentioned this alpha helical structure. So alpha helical structure was first time isolated. तो अपन normally बोलते हैं alpha, beta, delta, gamma. पहला जो उसका है उसका alpha. Now it looks like a helix. That's why alpha helical structure. Okay? Now second beta plated sheet. Alpha के बाद में क्या आता है beta. That's why beta. And ये क्या है? It will look like a plate of metal. It will look like a plate of sheet. That's why beta plated sheet. Okay? So beta plated sheet mostly that occurs between two peptide or say single peptide which is twisted back. So there is a difference between यहाँ पे hydrogen bonding होगा in between. Okay, the hydrogen bonding between two amino acids. Now tertiary structure of protein. Tertiary structure means three-dimensional arrangement of protein. Okay, in this again there is one introduction of new bond. There is introduction of hydrogen. There is introduction of one new bond known as a disulfide bond. Okay, so in addition to this CONH bond. CONH मतलब peptide bond. In addition to this CONH bond, in addition to this hydrogen bond, now in this tertiary structure of protein there is a disulfide bond. Then again in here ionic bonds are there. Van der Waals forces are there. Hydrophobic interactions are there. So in the tertiary structure of protein, so this type of bondings are involved. Hydrogen bonds, disulfide bond, ionic bond, van der Waals bond. तो आपको क्वेश्चन आ चुका है इसके ऊपर. Disulfide bond is the important property or characteristic of following structure of protein. तो disulfide bond किसमें होता है? tertiary structure में होता है. Secondary में hydrogen bonding होता है. Primary मतलब जो आपका peptide bond है वही. Are you getting or not? समझ रहे हैं आपको? Hello, am I audible? Because these bondings are very important. तो primary structure में आपका peptide bond होगा. Secondary structure में peptide bond plus hydrogen bond होगा. Okay, so this alpha helical structure is stabilized by only hydrogen bonding. Beta plated sheet is also stabilized by hydrogen bonds. Now tertiary structure, here peptide bond is also there, hydrogen bonds are also there, then disulfide bonds are also there, hydrophobic interaction, and ionic bonds are there, van der Waals attraction is there. So all type of bonds are present in the tertiary structure. Okay. Now what is quaternary structure? So when two or more polypeptide chains, now in the tertiary structure three-dimensional arrangement है. तो यहाँ पे प्रोटीन का मॉलिक्यूल कुछ ऐसा होगा, twisting and turning. Now in the quaternary structure two or more polypeptide held together by hydrogen bond, disulfide bond, again ionic bonds, hydrophobic interaction. One is the tertiary structure of protein. They are nothing but the levels of protein structure, primary, secondary, tertiary. So example of your tertiary protein, for example, hemoglobin. So in the hemoglobin you have four globin chains. Okay, you have four globin chains and four iron containing pigment known as a heme. है ना? hemoglobin में क्या होता है? चार ग्लोबिन चेन होते हैं, चार हीम यूनिट्स होते हैं. So that's why it will carry four oxygen molecule. ये अपने देखा है. So hemoglobin is there, quaternary structure. Then tertiary structure का आपका example, tertiary structure का quaternary structure का और एक example है insulin. Insulin is also structure of quaternary structure. Insulin में क्या होता है? दो polypeptide chain होता है. They are held together by disulfide bonds. Okay, so insulin. Now methods to determine the protein we have already seen. Secondary and tertiary structure of protein can be measured by X-ray crystallography. इसके ऊपर क्वेश्चन आ चुका है GPAT में recently, GPAT 2019 या 20 में. X-ray crystallography is used for the determination of secondary and tertiary structure of protein. Nuclear magnetic resonance NMR spectra give idea about the structural and functional information about the protein. Okay. So secondary, remember secondary tertiary structure of protein can be determined by either X-ray crystallography, okay, or NMR. Question आ चुके हैं इसके. Now you know that, okay, one amino acid, amino group of one amino acid and carboxylic group of another amino acid will interact to liberate water molecule, and there is a formation of peptide bond, CONH bond. So this bond is known as a peptide bond. C double bond ONH, right? CONH. This is a peptide bond. Now whatever this amino group is there, so here hydrogen is there, so another amino acid is there, so of which carboxyl group is there. So in between that there is a hydrogen interaction. That's why they are a secondary structure of protein. Secondary structure of protein में क्या होता है? हाइड्रोजन स्टेबलाइज करता है. हाइड्रोजन बॉन्डिंग are there. Hydrogen bonds are there in a secondary structure of protein. So you can see over here the alpha helical structure and beta plated sheet. So alpha helical structure you can see over here, okay, alpha helix. So in the alpha helix hydrogen bonds are there, okay. Hydrogen bonds are parallel to the polypeptide backbone. Hydrogen bonds are parallel to the polypeptide backbone. जैसे COH CO ग्रुप है उसका और NH3 का हाइड्रोजन बॉन्ड रहेगा. So a distance axial distance between amino acid is 1.5 angstrom. तो distance between two amino acid, ये जो दो amino acid होते हैं उसके बीच में का distance कितना है? 1.5 angstrom. So that is alpha helical structure of protein. That is the secondary structure, alpha helix. Okay, right-handed alpha helix is the most common. Left-handed is the least common. Then beta plated sheet. Okay, same peptide when it will twist, when it will turn. So there is a interaction between, again hydrogen interaction you can see over here. Hydrogen bonds which are perpendicular to the polypeptide backbone. इसके यहाँ पे perpendicular है, यहाँ पे क्या है? Parallel है. So hydrogen bondings they are parallel in case of your alpha helical structure, because helix में क्या है? Helix में so amino acids they are extending outward. यहाँ पे amino acid outward रहेंगे, तो यहाँ पे backbone क्या रहेगा? Peptide. amino acid side में रहेंगे. So due to that helix, so here in hydrogen bonds they are parallel in alpha helix, where in case of beta plated sheet they are perpendicular. Okay, इसमें axial distance between amino acid क्या होगा? 3.5 angstrom. इसमें ज़्यादा है. These are the two secondary structure of protein. You can see over here again alpha helical structure. So these are the hydrogen bonds, right? These are the hydrogen bonds. COO group जो रहता है उसमें और NH group जो है उसमें hydrogen bonds रहते हैं. So distance between each यहाँ पे 1.5 angstrom रहेगा. Distance between two amino acids 1.5 angstrom. And in each turn of alpha helix there are 3.8 amino acids. यहाँ पे रखना है. Beta plated sheet. So here also hydrogen interaction is there, but these are parallel. In case of hydrogen bonds are parallel in case of alpha helix. Here in case of beta plated sheet they are perpendicular. You can see over here again alpha helix. So alpha helical structure of protein, each turn of alpha helix जो turn होता है ये, जैसे मान लीजिए turn, Each ton of base is composed of a 3.6 amino acid. The distance between each amino acid is a 1.5 angstrom. Now, alpha helical structure is stabilized by hydrogen bonds. Hydrogen bonds. Hydrogen bonds. This one is known as beta plated sheet. Beta plated sheet. Beta plated sheet. Hydrogen bonds are perpendicular to primary structure of human insulin. Okay, primary insulin का structure कैसा होता है? Right? So insulin का structure, hemoglobin का structure, hemoglobin का तो already अपन देखा है. So there are 4 globin chains and 4 immunities. So in the insulin, now insulin is prepared. Okay, insulin is synthesized in the body from inactive peptide known as a pre-pro-insulin. Okay, there is a one peptide known as a pre-pro-insulin. यहाँ पे लिखता हूँ मैं pre-pro-insulin has a 110 amino acids. वहाँ पे 110 amino acids, 110 amino acids, 10 amino acids. So in the pre-pro-insulin, 110 amino acids are there. Now this is broken down. इस प्री-प्रो इंसुलिन में क्या होता है 110 अमीनो एसिड होते हैं. So pre-pro insulin 110 amino acid. So now it is broken down and converted into pro insulin. Converted into pro insulin. Converted into pro insulin. So which is again I think 76 amino acids. Okay? 76 amino acids. This is the structure of pro insulin. In the pre-pro insulin you have 110. This is structure of pro insulin. तो यहाँ पे 30 amino acid है. यहाँ पे 33 कितना हो गया 66. So 33 30 plus ये 21 66 plus 21 87 amino acid. So 87 amino acid is pro insulin. Now pro insulin has a 3 chains. Pro insulin has a 3 chain of peptide. This green chain is known as A chain, and this blue chain is known as B chain, and this red chain is known as C chain. C chain is also known as a connecting peptide. So A and B chain, they are connected by C chain in a pro insulin. Now after the modification, this C chain is removed and you will get your insulin. Now insulin has a 2 polypeptide chain. Polypeptide chain. This is known as chain A and this is known as chain B. Now in case of insulin, chain A has a 21 amino acid and chain B has a 30 amino acid. So insulin is having 51 amino acids. Insulin is having 51 amino acids. The conversion of pro insulin to active insulin. Okay. Now what this indicates? This is nothing but a site for proteolytic enzyme. जो proteolytic enzyme होता है यहाँ से break करेगा, ठीक है? और ये यहाँ से भी break करेगा. That's why this connector chain, which is also having a shape little bit like a C, and that's why it is known as a connecting peptide or connecting chain. So remember, in a pre-pro insulin 110 amino acids होता है. Pre-pro insulin में 110 amino acids होता है pre-pro insulin में, ठीक है? उसके बाद में pro insulin बनता है, उसमें 86 amino acids होता है. उसके बाद में insulin बनता है, इसमें 51 amino acids होता है. So 110, उसके बाद 86, उसके बाद है ना 51 amino acids. Are you getting or not? Now determination of primary structure. Are you getting? Am I audible to you? Determination of primary structure of protein. Determination of primary structure of protein. कैसे structure determine कर सकते हो आप? So first, determination of amino acid composition. Now proteins, they are hydrolyzed by either acid or by enzyme. Proteins are acid or enzyme hydrolyzed completely to liberate the amino acid, and these amino acids are quantitatively estimated. तो पहले उसका एनालिसिस करो अमीनो एसिड प्रोटीन का कंप्लीट और बाद में अमीनो एसिड एस्टिमेट करो. Now there is one enzyme mixture known as a pronase. Pronase is a mixture of protein that can completely pronase mixture of proteolytic enzyme which can completely hydrolyze the protein. Amino acids, they are separated, and they are separated by using chromatography, and then they are estimated. Now degradation of protein into polypeptide chain. जो प्रोटीन है ओरिजिनली जो कॉम्प्लेक्स प्रोटीन है उसको डिग्रेड करो into smaller polypeptide chain, into smaller polypeptide chain. Now protein is treated with the urea and guanidine hydrochloride to liberate the polypeptide. Now it it liberates the polypeptide by disturbing the non-covalent bond and dissociation of protein is there. ये क्या करता है गुआनिडीन हाइड्रोक्साइड नॉन कोवेलेंट बॉन्ड जो होते हैं हाइड्रोजन बॉन्ड आयनिक इंटरेक्शन उसको डिस्टर्ब करता है एंड दैट्स व्हाई देयर इज अ लिबरेशन ऑफ पेप्टाइड because in the quaternary structure in the tertiary structure you know that there is a two or more peptide polypeptide they are held together by hydrogen bonds. Now these hydrogen bonds they are broken by guanidine hydrochloride. So degradation कर सकते हो आप उसके बाद में फिर से पहला स्टेप करो और determine करो. Another region is there known as densyl chloride which will bind to N-terminus of the protein to identify the number of polypeptides. Polypeptides are broken down into fragments by enzymatic or chemical method. Now determination of amino acid sequence by Sanger region, Edmonds region, sequenter automatic machine is there. Sequenter automatic machine एक automatic machine होता है जिसमें आप सिर्फ प्रोटीन डाल दो, region डाल दो, वो आपको पूरा amino acid sequence बताएगा. Which determines the amino acid sequence. Now bonds present in the protein structure. ये तो already आपने देखा है फिर से एक बार. So first is a covalent bond. कौन से covalent bond होते हैं? Peptide bond is nothing but the covalent bond. Then disulfide bond is nothing but the covalent bond. So peptide bond and disulfide bond they are the covalent bond. So peptide bond is present in the primary structure. So disulfide bond is present in the tertiary structure of protein. So in the secondary structure there is only hydrogen bonds. Okay. Now non-covalent bond. Non-covalent bond कौन से है? Hydrogen bonds होता है, hydrophobic bonds होता है, van der Waals interaction होते हैं, है ना, electrostatic bonds होते हैं. They are the non-covalent bond found in the structure of protein. ठीक है. प्रोटीन बहुत ही बड़ा चैप्टर है अमीनो एसिड प्रोटीन. सो इट विल नॉट कंप्लीटेड इन 2 लेक्चर. सो ये अपना 3-4 लेक्चर तो मिनिमम लगता है, मिनिमम शॉर्ट में लिया तो प्रॉपर्टीज ऑफ प्रोटीन. प्रोटीन के प्रॉपर्टीज क्या है? सो ऑब्वियसली वो देखेंगे. नाउ they will form a colloidal solution with water. If you try to dissolve a protein in the water, so you will get a colloidal solution because of the hydrophobic, okay, hydrophobic, because of the hydrophobic नेचर ऑफ योर अमीनो एसिड्स. तो आपने अगर आपने अंडा जो है अंडे का जो प्रोटीन है उसको अगर वाटर में डाल दिया तो कोलोइडल सॉल्यूशन बनेगा. तो मोलिकुलर वेट ऑफ प्रोटीन दे डिपेंड्स. सो दे आर इन अ रेंज फ्रॉम 4000 टू 4000 to 4.40 lakhs 4.40 to 4,40,000 delta. Insulin has a molecular weight of 5700 delta. Myoglobin has a 1700 delta. हीमोग्लोबिन का मोलेकुलर वेट होता है 60 to 68,000 डेल्टा. ह्यूमन एल्बुमिन का वेट होता है ना 64 to 69,000 डेल्टा, है ना. सो मोलेकुलर वेट ऑफ प्रोटीन्स. प्रोटीन का शेप. नाउ देयर माइट बी डिफरेंट शेप्स ऑफ प्रोटीन. सो इंसुलिन हैज अ ग्लोबुलर शेप, एल्बुमिन हैज अ ओवल शेप, फाइब्रिनोजेन जो क्लॉटिंग फैक्टर होता है इट इज अ फाइब्रस और इलांगेटेड शेप. सो प्रोटीन का शेप भी अलग-अलग होता है. नाउ आइसोलेटेड पीएच ऑफ प्रोटीन. Now, as you know that there is isolated pH of amino acid, and proteins are nothing but the polymer of amino acid. प्रोटीन क्या है भाई? पॉलीमर है. तो अमीनो एसिड का एक आइसोलेटेड पीएच टाइप उसको क्या बोलते हैं अपन? द पीएच एट व्हिच अमीनो एसिड विल एग्जिस्ट एज अ न्यूट्रल आयन और अमीनो एसिड विल एग्जिस्ट एज अ बाइपोलर आयन known as a isolated pH, right? So protein also has the same thing. So isolated point of or isolated pH of protein means the pH at which protein has a least solubility. The pH at which protein has a least solubility or protein exists in a bipolar ion. Pepsin, that is a proteolytic enzyme, that is also protein, so it has a 1.1 isolated pH. Casein का होता है 4.6. Okay, so if you जो milk होता है milk, so if you store a milk, okay, and if there is a contamination in the milk, so this bacteria what will they do? They will decrease the pH of milk. They will decrease the pH of milk, and when pH of milk comes to 4.6, there is a breakdown of milk because milk is nothing but the emulsion. Milk क्या होता है? Emulsion होता है, right? तो वो breakdown होगा और क्या तैयार होगा? उसका coagulation होगा. And isolated pH, once there is isolated pH 4.6, there is a coagulation of casein, and that's why you will get curd. Albumin has a isolated pH of 4.7. Now urease enzyme जो होता है उसका isolated pH होता है 5. Hemoglobin का isolated pH होता है 6.7. The pH at which protein will exist in a two different ions by okay, two different ions, or at the isolated pH your proteins has a minimum solubility. कोई क्वेश्चन तो सेम ही रहेगा, अलग अलग तरीके से कुछ नहीं. So at the isolated pH, proteins are least soluble, or at the isolated pH, proteins they will get coagulated. Okay, are you getting or not? Are you getting or not? Yes or no? Am I audible to you? Audible आएगा. Precipitation of protein. Now, precipitation of protein, that is because of the different reasons. Now, protein can be precipitated by different way. So there might be dehydration or neutralization of polar groups. अगर प्रोटीन का पोलर ग्रुप जो है वो न्यूट्रलाइज किया, न्यूट्रलाइजेशन ऑफ प्रोटीन, पोलर ग्रुप ऑफ प्रोटीन, so that leads to precipitation. Precipitation at acidic pH also there. Precipitation by salting out. So if you add a salt, is very small amount of salt in a quaternary structure of protein, so that protein will be precipitated. Okay. Adding or dehydration by natural salt like ammonium sulfate or sodium sulfate. अगर सोडियम सल्फेट, अमोनियम सल्फेट अगर आपने ऐड किया क्वाटरनरी स्ट्रक्चर ऑफ प्रोटीन में, so what will happen? There is a salting out phenomena and there is a precipitation of that protein. Again, protein can be precipitated by adjusting to its isolated pH. Okay? Then protein can be precipitated by heavy metals like mercury, lead, zinc, chromium, cadmium. They can precipitate the protein. प्रोटीन को प्रेसिपिटेट कैसे भी कर सकता है. नो डीनेचुरेशन ऑफ प्रोटीन, इसके ऊपर पूरा एक पेज, एक नहीं 2 पेज दिया है सिर्फ ये टर्म प्रेसिपिटेशन ऑफ प्रोटीन इन द सत्यनारायण. तो सत्यनारायण का बुक देख लिया आपने, इसमें एक पूरा एक ही टर्म है प्रेसिपिटेशन ऑफ प्रोटीन, तो पूरा एक पेज दिया है. और डीनेचुरेशन ऑफ प्रोटीन मींस व्हाट? डीनेचुरेशन ऑफ प्रोटीन मींस लॉस ऑफ नेटिव स्ट्रक्चर ऑफ प्रोटीन. जो प्रोटीन का स्ट्रक्चर है वो लॉस हो गया तो उसको बोलेंगे अपन डीनेचुरेशन ऑफ प्रोटीन. Denaturation of protein. Now there may be because of the different method. Physical method is there. Denaturation मतलब आप हीट कर दो उसको, बल्ब चला दो, denaturation होता है. Chemical method is also there. Now denaturation of protein means loss of native structure of protein, which leads to denaturation. हो गया तो क्या होगा? Loss of the physical structure. Denaturation of protein result in the loss of physical structure, but native structure may remain intact. ना फिजिकल स्ट्रक्चर जो है उसका लॉस हो जाएगा, जैसे ग्लोबुलर है ना, यहाँ पे देखो शेप जो है इंसुलिन रेगुलर. सो लॉस ऑफ फिजिकल स्ट्रक्चर बट प्राइमरी स्ट्रक्चर में रिमेन इंटैक्ट. बिकॉज डीनेचुरेशन मतलब क्या है? डीनेचुरेशन मतलब सो ब्रेक डाउन ऑफ बॉन्ड्स, इंटरेक्शन जो है हाइड्रोजन इंटरेक्शन, आयनिक बॉन्ड, हाइड्रोफोबिक बॉन्ड, वो बॉन्ड ब्रेक हो जाएगा, डाइसल्फाइड बॉन्ड. देयर इज अ लॉस ऑफ नेटिव स्ट्रक्चर ऑफ प्रोटीन. CONH bond will be remaining, so that is native structure will remain, primary structure will remain intact. Native structure will lose and primary structure will remain intact. Loss of biological activity, जैसे प्रोटीन है आपका इंसुलिन. इंसुलिन क्या है? एक प्रोटीन है, उसका काम क्या है? To decrease the its blood sugar level. So if you heat the insulin, if you denaturate the insulin, so there is a loss of biological activity of insulin. Also, denaturation can result in the insolubility, increase in the viscosity. जैसे for example, आपका अंडा है, अंडा बड़े खाता हो ना आप अंडा. If you see in the egg you have protein, liquid होता है, liquid ना. If you heat that or if you boil the egg, boil किया आपने egg नहीं, so क्या होगा? Coagulation of protein. So increase in the viscosity, okay, result in the insolubility. ये insolubility है, मतलब solubility कम हो जाएगा, decrease in the solubility, loss in the biological activities. That is a denaturation protein can result in the denaturation of protein, increases the ionizable group in the protein and sulfhydryl groups in the protein, which can be easily digested. So denatured protein can be easily digested. That's why आप क्या करते हो? कच्चा थोड़ी खाते हो, कच्चा अंडा थोड़ी खाते हो. तो खाते जो रेसलर्स होते हैं, पहलवान होते हैं, वो खाते हैं कच्चा अंडा, रॉ एग. They used to eat raw egg. But we humans, we used to denaturate the protein present in the egg and then we used to eat. Because denaturation increases the ionizable group or sulfhydryl group. Now this protein can be easily digested. Denaturation are usually irreversible. Denaturation जो होता है usually irreversible. एक बार डीनेचुरेट हो गया तो प्रोटीन का स्ट्रक्चर फिर से आप रिगेन नहीं कर सकते. रिमेंबर, डीनेचुरेशन is usually irreversible. Careful denaturation sometimes can cause reversible, and reversible denaturation is called as renaturation. रिवर्सिबल डीनेचुरेशन जो होता है उसको डीनेचुरेशन, उसको रीनेचुरेशन भी बोलते हैं. नाउ हीट कोएगुलेशन टेस्ट इज कॉमनली यूज्ड टू डिटेक्ट द प्रेजेंस ऑफ अल्बुमिन इन द यूरिन. है ना, आपके यूरिन में अगर अल्बुमिन है तो कैसे चेक करेंगे? यूरिन का सैंपल ले लो टेस्ट ट्यूब में, थोड़ा सा उसको बर्नर के ऊपर हीट करो. अगर उसमें turbidity आता है, if urine become turbid, that means there is a presence of albumin in the urine. So heat coagulation test is commonly used to detect the presence of albumin in the urine. क्यों? क्योंकि that causes the denaturation or that causes the denaturation of protein and decreases the solubility. तो याद रखना है, right? So proteins after denaturation. You can see the structural hierarchy of your protein again. So level of protein organization in the level of organization of protein structure. Okay, now this is known as a primary structure. Primary structure का है simple, this is simple amino acid chain, है ना, amino acid का sequence रहेगा यहाँ पे, and that is known as a simple. So primary protein structure, primary structure of protein, it is a sequence of amino acid chain, amino acid in the protein. Now secondary structure of protein. So in the secondary structure of protein, so in the primary you have CONH bond, that is a peptide bond. Secondary structure of protein में क्या रहेगा, which occurs when sequence of amino acid are linked or joined by hydrogen bonds. Okay, so alpha helical structure is there and beta plated sheet is there. So these are the two secondary structure of protein. Question ऐसा आता है आपको, alpha helical structure of protein belongs to following type. प्राइमरी स्ट्रक्चर ऑफ प्रोटीन, सेकेंडरी, टर्शियरी, क्वाटरनरी. सेकेंडरी प्राइमरी स्ट्रक्चर में सिर्फ पेप्टाइड होगा, सिंपल अमीनो एसिड चेन, सिंपल अमीनो एसिड पेप्टाइड, सिंपल अमीनो एसिड सीक्वेंस. सेकेंडरी स्ट्रक्चर में होगा हाइड्रोजन बॉन्ड between the amino acids. If the peptide is twisted and turned, twisting is there in case of secondary structure. There are two types of secondary, alpha helix and beta helix. Now tertiary structure of protein. Tertiary structure of protein. Tertiary structure, when it occurs when there is a two or more peptide will have interaction. When two or more peptides will have interaction through hydrogen bonds, hydrophobic bonds, ionic bonds, and disulfide bonds. So there is a tertiary structure of protein. Okay. Now what is quaternary structure? Again there is a folding and defolding. So when protein consisting of more than one amino acid chain, इसमें दो तीन polypeptide chain क्या होगा, साथ में होगा. So this is nothing but the different structure of protein: primary, secondary, tertiary, and quaternary structure of protein. Now different bonds are there in a primary structure, secondary structure, and tertiary structure. Primary structure में क्या होगा, यहाँ पे पेप्टाइड बॉन्ड होगा. सेकेंडरी में ये, in the secondary structure, peptide bond plus hydrogen bonds. In the tertiary structure, peptide bond, hydrogen bond, hydrophobic interaction, van der Waals process, ionic bonds, disulfide bonds. And in the quaternary structure, so two polypeptide chains, they are held together by hydrogen bonds again. यहाँ पे hydrogen bonds है. बाकी tertiary structure का bond तो रहेगा यार एक peptide में, but यहाँ पे two different peptides, they are held together by hydrogen bonds. तो याद रखना है आपको, different bonds in the structure of protein. Question आता है इसके ऊपर. Again we will see next part. Next is nothing but the classification of proteins. Classification of proteins. Proteins, they are classified. So simple proteins, conjugated proteins, and derived proteins. So what do you mean by simple proteins, conjugated proteins, and derived proteins? Simple proteins, because simple proteins, they are also classified into two types. First one is a globular proteins. So in that आपका यहाँ पे एग्जांपल होगा albumin, globulins, gluteins, prolamines, histones, globins, protamines, and lectins. Now scleroproteins, they are the structure. उनका सब scleroproteins, they are present in the structures. They are the building bricks body of body. So collagen is there, elastin is there, keratin are there, right? Now conjugated protein मतलब क्या? So some other part is there. Conjugated protein मतलब some other part is there. So nucleoprotein means what? So protein plus nucleic acid. And nucleoprotein is protein plus nucleotides. Glycoprotein is what? A conjugation of protein with the sugar. Mucoprotein conjugation of protein with the N-glucosamine. Lipoprotein means what? A macromolecular complex of lipid with the protein. Phosphoprotein मतलब क्या? Phosphoric group, phosphoric acid रहेगा उसमें. Chromoprotein is है ना यहाँ पे chromoprotein. Metalloprotein is complex of protein with the metal. जैसे for example hemoglobin. What is hemoglobin? It is a metalloprotein because in that you have ferrous ion. Metalloprotein. Now derived protein are nothing but they are the derivatives of derivatives obtained from the simple or conjugated proteins. So in that you have two primary derived proteins and secondary derived proteins. So primary derived proteins. So here coagulated proteins and metaproteins. In the secondary derived proteins में आपको proteose, peptones, polypeptides and peptides. They are the breakdown product of your simple protein or conjugate protein. So this is just classification. और भी इसमें डिटेल में पढ़ना है तो आप क्या पढ़ सकते हो इसमें और या फिर इंफॉर्मेशन कलेक्ट करना है इसमें का तो और क्या इंफॉर्मेशन कलेक्ट कर सकते हो आप सो जैसे एल्बुमिन के बारे में सब कुछ पढ़ लो ग्लोबुलिन्स के टाइप्स पढ़ लो ऑलरेडी हो चुका है वेयर इज अ प्रोलामाइंस किसमें होता है स्टोन्स किसमें होता है ये स्टोन्स मोस्टली दे आर द बाउंड इन द न्यूक्लियस देन कोलेजन के बारे में देख लो लाइकवाइज दैट नाउ ओवरव्यू ऑफ मेटाबॉलिज्म ऑफ अमीनो एसिड मेटाबॉलिज्म जो होता है उसका ओवरव्यू क्या है overview of metabolism of amino acid. Now you have particular storage of amino acid in your body. Your body has a particular store of amino acid known as amino acid pool of your body. The body में amino acid store होता है. Free amino acids are present in the body. Around 100 gram of amino acids they are present in the body. 100 gram of amino acids they are present in the body. They are known as the amino acid pool of your body. Amino acid pool of your body. Now from here this amino acid comes ये जो बॉडी का अमीनो एसिड का पूल है वो कहाँ से मिलेगा अमीनो एसिड आपको जो आप दबा दबा के खाना खाते हो बुक्कर के जैसे बुक्कर के जैसे आप खाना खाते हो डाइटरी प्रोटीन अराउंड 50 to 70 ग्राम ऑफ प्रोटीन डेली यू आर ईटिंग so from that after hydrolysis you are going to get amino acids now from breakdown product of your body's protein आपके बॉडी के खुद के प्रोटीन जो है वो उसको ब्रेक डाउन करती है बॉडी so near about 300 to 400 gram protein they are degraded per day that's why you need a now from that you are going to get amino acids now what is amino acid or these amino acids will be utilized for the जो भी amino acid pool है आपके बॉडी का या फिर जो भी आपको amino acid मिलता है डाइट से तो उसका यूज क्या होगा उसका कहाँ पे जाते हैं उसका और मेटाबॉलिज्म right so from these amino acid so they are metabolized amino acid they are metabolized into keto acids and ammonia is released after metabolism of amino acids जो भी मेटाबॉलिज्म होगा उसमें कीटो एसिड बनेगा और अमोनिया बनेगा अमोनिया जो बना है अमोनिया इज टॉक्सिक टू योर बॉडी अगर फ्री अमोनिया बढ़ गया आपके बॉडी ब्लड में आपको चक्कर आना चालू हो जाएगा इट इज अ न्यूरोटॉक्सिक एंड आपका लिवर भी खराब हो जाएगा किडनी खराब हो जाएगा नाउ when there is an increase in the ammonia level of your body when you have liver problem if there is a cirrhosis of liver cirrhosis हो गया आपका आपने क्या किया आप बन गए कबीर सिंह बन गए आपने पीना चालू किया डेली बॉटल खाली कर रहे हो क्या होगा आपका लिवर खराब हो जाएगा लिवर cirrhosis and because of that liver will be the capacity of liver or the metabolic capacity of liver will be decreased day by day urea cycle okay so कम हो जाएगा that's why you will get accumulated urea will get accumulated you will get in a coma okay तो जो जिसका लिवर खराब है उसका यूरिया लेवल बहुत ही बढ़ता है जिसका किडनी खराब है किडनी will not excrete urea that's why there is increase in the urea so near about daily 15 to 30 gram of urea which is being excreted per day so ammonia will be converted to urea through urea cycle and this urea will be excreted. Now, amino acid can be used for oxidation purpose to get energy. Now, from amino acid, so there is synthesis of glucose, there is a gluconeogenesis. From amino acids, again, synthesis of non-essential amino acid will be there. From amino acids only, amino acids only. Synthesis of specialized different important body product, like there is series of heme, purines, pyrimidines, creatinine, enzymes and hormones. From the amino acid only, synthesis of body proteins. Whatever proteins are required in the body, that will be synthesized through amino acids. So what will happen with the amino acid pool? So around 100 gram of amino acids, they are stored in the body. तो ये amino acid के साथ में क्या क्या हुआ, क्या क्या हो सकता है कि amino acid के साथ में क्या क्या हो सकता है, आता कहाँ से, आने का सोर्स ये है, जाने का सोर्स क्या है, ये सब जाने का सोर्स है, आने के लिए कहाँ से आता है, जाने के लिए कहाँ पे है, और रिजर्व स्टोर क्या है 100 ग्राम. So it is an overview of metabolism of amino acid. Now, if you see the metabolism of amino acid, so every individual amino acid you can see. प्रत्येक every amino acid का अलग से मेटाबॉलिज्म होता है. ग्लाइसिन का मेटाबॉलिज्म अलग से हुआ, फिनाइलएलानाइन जो एरोमेटिक अमीनो एसिड है उसका मेटाबॉलिज्म अलग से हुआ, है ना, जो एसिडिक अमीनो एसिड है उसका मेटाबॉलिज्म अलग से. सो एवरी अमीनो एसिड इज मेटाबॉलाइज्ड सेपरेटली. ओके, सो इफ यू सी, अगर उनको प्रॉपर्ली समझ के सब कुछ पढ़ना है, so it will take a lot of time, so it is not easy to cover the metabolism of every single amino acid. Okay, but whatever I can, whatever is important I will explain. Are you getting or not? समझ